All right, so we just finished up taking a look at chromosomal mutations. Chromosomal mutations are massive changes to a whole bunch of DNA on a chromosome rather than just a little bit. We're looking at point mutations now. Point mutations are tiny little changes to DNA. So if you have a whole stretch of DNA, it would be UV light, it'd be carcinogens, it'd be something coming in and just changing this red to a purple, or this purple to a blue. Okay, it's just like a little point mutation, we call it. One little DNA base or nucleotide is changed relative to chromosomal mutations, which is a big chunk, a big section of DNA, or a big section of the chromosomes changed. Okay? So one or more nucleotides are inserted, deleted, or changed. The result can be that the polypeptide is wrong, right? So instead of having certain amino acids in here, one or many more amino acids could be different. Now again, what's the outcome if there's a mutation? There's 3 outcomes. Not of point mutations, but just of mutations. What's the 3 outcomes again? Positive, negative, neutral. Okay, we don't know if it's going to be good. Mind you, we do know that what we got is okay. So we try to limit mutations if we can. And that's why we would suggest strongly that you put on sunscreen, right? Because, yeah, sure, there might be a positive mutation out of that. But our experience is that usually it's a negative mutation, overexposure to UV. Now, there are 4 different types of point mutations. A silent point mutation, a missense point mutation, a nonsense point mutation, and a Frameshift point mutation. And you have them on your worksheet there. Okay? So this one here, normal gene, silent mutation, missense, nonsense, and Frameshift. And there's space to put a definition and all that stuff. And you can see what the outcomes are. So let's start with a normal gene. So very top, this is what we're going to put in. If you have a DNA sequence of GGG, CCC, TTT, AAA, and along comes RNA polymerase, which has always been this paperclip, and it produces an mRNA sequence. Yoink. What is the sequence on the mRNA? So on that top one, GGG becomes CCC. CCC becomes, it's transcribed as GGG, and TTT is transcribed as AAA. And finally, AAA is transcribed as UUU. Now, because we're geneticists, right, after one day of learning about this, we pull out our genetic code and we translate it now. Translating CCC is what? Proline. PRO, if you like. GGG. Glycine, if you have that in front of you, you'll see it. It's glycine. GLY. AAA. Lysine. LYS. And last but not least is UUU. It's got a long name. Phenylalanine. Phenylalanine. PHE, if you like. Good. So that's the normal sequence. I'm not saying it's a good sequence, not a bad sequence. That's just a normal sequence in our DNA. Okay? So there you go. Proline, glycine, lysine, phenylalanine. What we're going to do is we're going to make a change. So UV light, you smoke cigarettes, right? You sleep with asbestos or uranium, I don't know. But there's a point mutation. And here comes the point mutation. Doink. See what happened there? The G became GGC. There's a point mutation. One point was changed. One base was changed. The G changed to a C. So you can put that down right there. You can say the mutated G sequence is now GGC and still CCC, TTT, AAA. That means our RNA sequence, our mRNA sequence is now CCG, GGG, AAA, UUU. Does it stay proline, glycine, lysine, phenylalanine? Well, let's see. I think the last three are okay, but what is CCG code for? Still proline. This is a silent mutation because although there was a point mutation, it doesn't change the amino acid sequence. So however this thing worked, it still works exactly the same way. Okay? We don't know if it's a good gene to have or a bad gene to have or whatever, but it works as it usually does. Okay? So that's called a silent mutation. I think the name makes sense. The next one here is going to be a missense mutation. So again, we're going to start with this, but this time, this radiation comes in, and it's going to change this T, this third T, into an A. Okay, so one thing is going to change. That bad boy right there is going to change to an A. If that changes to an A, the mRNA sequence is not AAA. What is it? It's AAU. Let's hope that lysine is there. AAU. Because if AAU is lysine, then it's a silent mutation. AAU. What? It's asparagine. So, is this the same? Proline is the same. Glycine's the same. Phenylalanine is the same. There's a single amino acid change. So that is a missense right there, asparagine. Now, again, do we know what the outcome is? Do we know if this is good, bad, or neutral? Nope. We're going to have to see. This could be the one that gives you superpowers, right? You could run through walls. This could be the one that makes you have a tail. This one could make you actually function exactly the same way as you are now. So we don't know. A good example of this one, a single point mutation causing a change in the amino acid sequence. This is red blood cell. The term for it is biconcave because it looks like a donut and you just squeeze the middle, but you didn't punch a hole. So there's the concave here, cave here, biconcave. And this is perfectly round, and the concaveness makes it so that when it travels through those really narrow blood capillaries, it can fold and fit through and come out the other end. It holds on to oxygen. Sometimes it holds on to carbon dioxide as well. We'll find that carbon dioxide travels better a different way, but it still travels through the blood, but not on your red blood cell. But you can suffer from a disorder called sickle cell anemia. And sickle cell anemia, I don't know if you can see that too clearly, but this one looks like that one there, and this one here, and that one here. And then some of them have this pointy shape to them, like a sickle, right? If you think of the Russians, the communists, and they have the sickle. Death carries a sickle, right? When he walks around with his staff with the blade on the end. Those are not very effective at carrying oxygen. In fact, your body will destroy them. But they're there, and they occupy space. And as a result, you don't transport oxygen very well. So you suffer from anemia. And this one's not because you didn't eat enough red meat or that you lost blood because of a traumatic stabbing or something. This is because you have funny-looking red blood cells as well as normal red blood cells. So that was a simple nucleotide difference. It's supposed to be GAG, and instead of having GAG in this gene for hemoglobin, you have a GTG. So A turned to G. Next one, nonsense. This one codes for a stop codon. So there we go. That's our regular one. And I'm going to change it so it's now ATT. See, single point mutation. Everyone agree? One little point, poke it in the eye, drag it out, and throw another one in there instead. Happened because of UV light, happened because of a chemical mutagen, right? A carcinogen. Could have had small chance, could have been a viral component that changed it, a vector. But anyways, it became ATT. That means when my ribosome comes in there, sorry, my RNA polymerase, my apologies, my RNA polymerase comes in here, instead of making our RNA AAA, it's going to be what? UAA, thank you. And that UAA, if you take a look on your genetic code, UAA is one of the 3 stop codons. Stop. Now here's the deal. This has a whole bunch of dots behind it. That means there's a whole bunch of more DNA. That means there's a whole bunch more RNA. That means there's probably a whole bunch more amino acids. Now it could be just a handful. It could be 10 or 100 or 1,000. It could be tens of thousands of amino acids. This could have happened right at the start. So does it affect the protein? Yes. Does it affect the result of the protein, how it works? Yes. But do we know if it's good, bad, or neutral? No idea. Right? So this one, I think you can see, could be quite profound, especially if it's right at the beginning of the gene, right, or near the middle. But if it's near the end, there could be just a little bit of its tail cut off, and it works just as fine. Cystic fibrosis and muscular dystrophy are 2 examples of it. Okay, so this is a mutation to the CFTR gene. So for cystic fibrosis, and you get thick mucus, and lung infections. You have to take these drugs to thin out your mucus. Physical manipulation too. People have to pat you on the back to loosen it up on your throat and stuff, and then you hork it up. Your lung, you eventually are drowning in your own mucus, and your life expectancy is quite short relative to a normal person with a normal lung capacity. Right? Crohn's disease has been tied to it. So if you know someone with ileitis, colitis, Crohn's disease, right, that's been tied to it. And muscular dystrophy, a mutation in the dystrophin gene. So that's a structural component of the muscle tissue. So that would be negative. Frameshift mutation.